In this video, I'm going to demonstrate the gram stain procedure. The first thing we need to do prior to gram staining is to make a smear from the culture that we prepared. In this image, you can see my gloved hand holding a tube that contains culture material. Notice the aseptic technique. The tube is being held at an angle, and the cap is being held in my fingers. My other hand, which is outside of the image, is holding a sterile inoculating loop. The inoculating loop gets inserted into the culture container so that the loop comes down to about halfway in the volume of the culture. With that loop full of culture material, I'm now ready to make my smear. Notice that I've placed the loop down in the center third of the microscope slide. I'm going to rotate the loop around in a circular motion, spreading out the culture material over that central third of the slide. Notice that I'm starting in the center and I'm moving outwards. Be sure to spread the culture material across that entire 1/3 central area of the slide. The more you spread the material out, the faster your smear will dry. Notice too that the microscope slide has been labeled. Don't forget to always label your slides. Once the smear is completely dry, we're ready to heat fix the cells onto the slide. Remember, we never heat fix a wet smear. And that's because if we applied a wet smear to a source of heat, like the incinerator seen in this image, there's a possibility that we would vaporize the fluid and send bacterial cells into the air in the laboratory, which would be hazardous to ourselves and to our colleagues. So once the smear is completely dry, attach a clothespin to one end of the slide and then place the slide on the aluminum housing surface of your hot incinerator. Remember, these incinerators get very hot. The purpose of the clothespin is to keep our fingers as far away from that hot surface as possible. Lay the slide down onto the aluminum housing, and in your head, count to about 20 seconds. This should be plenty of time to heat fix the cells onto the slide. It's not uncommon for us to prepare multiple smears at the same time when we're working in the laboratory. When you're performing the gram stain procedure, prepare your slide and your partner's slide first before you move on to staining. We make use of staining trays in the microbiology lab. These trays are very low tech, made out of plastic food containers and zip ties or coffee stirrers. These zip ties in this image provide a platform for us to lay our smears. In this way, when we lay down our smears and apply stain, any excessive waste stain will go down into the staining tray and not onto our desktop. Remember, the stains that we use in microbiology are toxic. We cannot put them down the drain. Instead, they have to be sent away as hazardous waste for correct disposal. Gram stains are often provided to us in a kit form. On this slide, you can see the 4 liquids that we're going to be using in the gram stain procedure. We have crystal violet, which is the first stain that's applied to the smear. We have Gram's iodine, which is our mordant. we have our decolorizer solution, and we have safranin, which is our counter stain. Apply the crystal violet across the slides, making sure that you have covered the entire area of the smear. Crystal violet is not a particularly expensive stain, so make sure that you place a big puddle of stain that completely covers that smear. You don't want to miss any cells that are on your smear. The crystal violet is allowed to sit for 30 seconds, and then we lift the slides one by one and gently rinse off the extra stain with distilled water. Allow all of the extra stain and all of the rinse water to go down into your staining tray for later disposal. Notice that on this rinsed slide, I can't see anything in the area of my smear. You will not see any purple color on your smear because gram stains stain the cells, not the slide. And the cells are microscopic. We're not going to see any color until we go to the microscope. The next chemical that is applied to the smears is Graham's iodine. Graham's iodine is a mordant, and that means that it's used to help the crystal violet stain penetrate and adhere to the peptidoglycan in the cell wall. Graham's iodine also needs to be applied all the way across the surface of our smear. Make sure that you put a puddle of Graham's iodine on top of your slides. Similar to the crystal violet, Graham's iodine stays in place for 30 seconds. One at a time, lift the slides with a clothespin and hold them at an angle over your staining tray so that the excess Graham's iodine can drain into your staining tray. Then be sure to rinse your smear with distilled water. The next step in the Graham stain procedure is the only step that requires strict timing, and that's the decolorization step. Graham decolorizer is an alcohol solution, and we're going to apply it to the smears not as a puddle, but as a drop-by-drop process that will help remove some of the crystal violet from the peptidoglycan in the cell wall. This is the only step where you have to be very careful with your timing. On this slide, you can see that I'm holding the smear over the staining tray. I've drawn up some of the decolorizer into a transfer pipette, and I'm ready to go. I'm going to apply the decolorizer drop by drop so that it runs down over the smear, and I'm going to do that for 10 seconds. Apply the decolorizer drop by drop so that it runs over the surface of the smear for 10 seconds. As before, you're going to want to rinse off the decolorizer after this procedure. Hold the slide at an angle over the staining tray and gently rinse the smear with distilled water. Don't forget this step. If you skip the rinse step here, the decolorizer will stay on the smear for much too long. Now we lay the smears back in the staining tray so we can apply the safranin. Safranin should be placed in a puddle over the entire surface of the smear and left this time for 60 seconds. The safranin should be rinsed off with distilled water just like all the other liquids that we applied. At that point, the slides are ready to be examined. Gently blot any excess fluid off of the slides by pressing down with blotter paper. It's not important that the slides are completely dry. You just want to remove the majority of the rinse water that's left behind. You can see that these slides are still a little bit wet, but that's okay. It won't interfere with our ability to examine them under the microscope. Notice too that the slides look kind of messy. You're going to get some staining of the slide itself during a gram stain procedure. That really can't be helped. What's important though is what's going on in that central third of the slide where we placed our smear. We're going to take these to the microscope and we're going to focus in this area to find our cells. Remember, before you put the slides on your microscope, be sure to remove your gloves. Gram staining is done so that we can determine the gram status of bacterial organisms. On this slide, you're looking at the view of a bacterial smear under the 100x oil immersion lens. So we are examining these cells at a total magnification of 1000x. Notice that there are two cell types on this smear. There is a smaller circular cell, and there's a larger elongated cell. The circular cell, or the cocci, have stained a dark purple, and the elongated cell, or the rod, has stained pink. In gram staining, a purple color tells us that the cell is gram positive, and a pink color tells us that the cell is gram negative. So from this smear, I would conclude that I have a gram positive coccus and a gram negative rod.